Alright guys, today what we're going to do is we're going to talk about translation to finish up our concept of protein synthesis. So I want to start by going over the text, and then we'll use the video on the side to help you understand what the text is actually saying. So translation. The sequence of nucleotide bases in a mRNA molecule serves as instructions for the order in which amino acids should be joined together to produce a polypeptide. However, anyone who has tried to assemble a complex toy knows that instructions generally don't do the job themselves. They need something to read them and put them to use. In the cell, that something is a tiny factory called ribosomes. The decoding of an mRNA message into a polypeptide chain / or protein is known as translation. Translation takes place on the ribosome. During translation, the cell uses information from messenger RNA to produce proteins. Refer to figure 12-1218 as you read about translation. Step A. Before translation can occur, messenger RNA must be first transcribed from DNA in the nucleus and released into the cytoplasm. So if you guys look at the video here, okay, here's a cell, there's the nucleus, and just like it says here, the first step is that the RNA must first be transcribed. So in the nucleus, as you can see, we have chromosomes. Chromosomes are just, DNA that's been tightly coiled together, and because it's super long. And so whenever we need to make a protein, there's a specific segment of DNA that has codes for making that protein that the cell needs. So in this case, this big long portion represents that piece of DNA that we're going to use. That has the instructions to make the protein. And so what you're seeing here is what the text is referring to as transcription, right? So the DNA is being used to transcribe. The DNA is being transcribed into RNA, and so as you can see, the RNA is synthesized there by the RNA polymerase. Okay, once the RNA is complete, it gets edited. The introns get cut out, the exons get spliced together, and the mRNA molecule exits the nucleus. So that's basically what it's saying there in step A. Translation begins when an mRNA molecule in the cytoplasm attaches to a ribosome. As each codon of the mRNA molecule moves through the ribosome, the proper amino acid is brought into the ribosome and attached to the growing polypeptide chain. So let's take a look. So here's the mRNA molecule, and notice, like it says, okay, it's in the cytoplasm now, and it attaches to a ribosome. So there's the ribosome, and so now the ribosome attached to the messenger RNA molecule, just like it said in the text. The ribosome does not know which amino acid to match to each codon. So remember that mRNA has codons, those 3 consecutive nucleotides, and so that's the job of the transfer RNA. Okay, so the transfer RNA are the ones that are going to match bring the amino acids and match them to the correct codons. So here's a tRNA, there's the amino acid, and remember the bottom piece here, okay, that is the anticodon. Okay, UCA is the anticodon here. And so what happens is the tRNA brings the amino acid, and if you notice UCA, okay, that's a match to AGU, to this codon here, right? And so these anticodons and codons have to match. If they don't match, then the ribosome won't connect the amino acids that are attached to each other. So let's continue. So that's the job of the RNA. Each tRNA molecule has an amino acid attached to one end and a region of 3 unpaired bases to the other. The 3 bases on the tRNA molecule are called anticodons. A complementary they are complementary to one of the mRNA codons. So that's what they're talking about here. So we're talking about, if we drew this here, okay, this right here, that's the anticodon, and it's complementary, okay, to the codon here. So this is the codon, right? And so that's what it means. That it complementary, there's a match there. The bases are able to join together, and that basically is what ensures that that allows the ribosome to connect the amino acids. Okay, if there's no match, then the ribosome won't accept those tRNA molecules that come in. So let's continue. In the case of the tRNA molecule for methionine, the anticodon bases are UAC, which pair with the methionine codon AUG. The ribosome has a second binding site for a tRNA molecule for the next codon. If the next codon is UUC, a tRNA molecule with an AAG anticodon would fit against the mRNA molecule held by the ribosome. The second tRNA molecule would bring the amino acid phenylalanine into the ribosome. So if you take a look here, okay, that's what it's referring to. So you have tRNAs, right? And so here in this particular mRNA, right, you have AUG. Okay, so here's AUG right here. Okay, all right, and so the tRNA comes in, okay, that's carrying phenylalanine, and because the anticodons match, right, then the tRNA binds to the anticodon binds to the codon, right, and so then the ribosome has a second slot, right, and so here comes phenylalanine, right, attached to this anticodon, and or to this tRNA, and these tRNAs only carry these specific passengers. So you won't ever see this tRNA carry any other any other amino acid. This anticodon with AAG will always carry phenylalanine, okay, and it will always attach to this codon here, UUC. And if you look at our codon table, if you look up UUC, you'll notice that phenylalanine is the amino acid that it codes for. So now that it has these two slots filled, okay, then the ribosome can now initiate its work. So let's go ahead and continue. Like an assembly line worker who attaches one part to another, the ribosome forms a peptide bond between the first and second amino acids, methionine and phenylalanine. At the same time, the ribosome breaks the bond that had held the first tRNA molecule to its amino acid and releases the tRNA molecule. The ribosome then moves to the third codon where a tRNA molecule brings it the amino acid specified by the third codon. So this is what it's saying here. Okay, so if you take a look, um, we have here is that tRNA that was carrying the green amino acid phenylalanine or methionine, I'm sorry. And so what happened is the ribosome attached these two guys together, right? Okay, and then once these two got attached, okay, it broke the bond here, okay, to this guy, and now this guy gets released. Okay, it's like a taxi or an Uber dropping off its passenger at the airport, right? And so once it drops off its passenger, it goes and it's released and it goes to the cytoplasm where it's going to pick up more methionine, so it can continue to deliver these methionines to the ribosome. And so now here's the first codon, here's the second codon, and now here's the third codon. Okay, and so in the reading it says that once the first and second amino acids gets connected, right, and the first amino acid gets disconnected from its tRNA, now the ribosome continues to translate the mRNA, moves to the third codon, which is AAA, and this tRNA comes in with the anticodon UUU carrying lysine. And so now the ribosome will connect these two okay, amino acids to this one, okay? And the process will continue. So if you look at the video here, you can see this in process, right? So see, every time a tRNA comes in, it matches it to a codon, okay? And anticodons, codons match. And every time there's a match, the ribosome continues to connect these amino acids to the next amino acid that comes in. And so this essentially here is what they're referring to as the polypeptide chain. In class, we've been saying protein, and because I don't want to get you too confused, but basically, most proteins are made up of multiple chains of amino acids. But for our class, just think of a protein as a single chain of amino acids, and once this protein is completely built, okay, or this polypeptide chain is completely built, okay, it will get released, and the protein will go do its job. Okay, so let's continue with the reading and see where we're at. Okay. The polypeptide chain continues to grow until the ribosome reaches a stop codon on the mRNA molecule. So as you can see, here's the polypeptide continuing to grow. Now, notice these amino acids will continue, okay, to be brought by tRNAs until, okay, the ribosome, okay, finds a codon that tells it to stop doing the protein synthesis, right? So when the ribosome reaches the stop codon, it releases the newly formed polypeptide. And remember, a polypeptide is just a chain of amino acids. So for us, it's just a polypeptide. Just think of a protein, and the mRNA molecule completing the process. So as you can see in the video, okay, the polypeptide chain is growing, or the protein is being made, right? And when it reaches that stop codon, the polypeptide, okay, is released. And what you need to know about proteins is that, okay, that chain of amino acids, okay, the sequence of amino acids, okay, so the sequence in which those amino acids were connected determines the shape of this protein. And the shape of the protein, okay, that shape determines the job of the protein. Okay, now you might be thinking, what the heck, Rodriguez, what are you talking about? Well, it's kind of like this, guys. Okay, it's not very hard to understand if you follow along. So for example, if I had two tools and one tool looked like this, okay, this probably be a screwdriver, right? And if I showed you another tool that looked like this, a hammer. Now, what do you notice about these two tools, right? These two tools, right? They have different shapes, right? And the shape determines the job of the tool. So it's the same concept with proteins. Proteins have a specific shape, and that shape is determined by the way those amino acids were connected. Okay, that's why the instructions are so important, because the instructions tell the ribosome in what sequence to connect amino acids. Once that protein is made, those amino acids, okay, interact with each other, and the protein folds like this. And that folding, okay, is going to determine, it's going to give it its shape, right? And the shape is going to determine its job. Okay, so proteins that have different amino acid sequences will have different shapes and therefore have different jobs. Okay, so it's very important that you understand that concept. That's probably one of the most difficult concepts for biology students to understand at any level, not just high school level, but even college level. So it's very important that you guys understand this concept. Okay. Okay. So now let's finish up the reading here. So roles of DNA and RNA. You can compare the different roles played by DNA and RNA molecules in directing protein synthesis to the two types of plans used by builders. A master plan has all the information needed to construct a building, but builders never bring valuable master plans to the building site where it might be damaged or lost. Instead, they prepare inexpensive disposable copies of the master plan called blueprints. The master plan is safely stored in the office, and the blueprints are taken to the job site. Similarly, the cell uses the vital DNA master plan to prepare RNA blueprints. So that's very important there, right? So they're comparing the building of a protein to the building of any structure or a house or a building and so forth. So you got to remember that DNA is the master plan and RNA are the blueprints. They're the copy of that master plan. Okay, and so if you take a look at our video here, remember where those plans were first copied, right? So here are all the master plans, okay, for building the protein that the cell needs, right? They're all in this DNA. And so whenever it needs to build a specific protein, it goes to that part of the DNA that has the instructions and it copies that piece of instruction. It doesn't copy the whole thing, it just copies that one particular piece. Like, for example, maybe this piece of DNA here, okay, right here, maybe this piece of DNA, all right, this gene, maybe that's for a protein that's going to give you your specific eye color. And maybe this portion over here is a different piece of DNA, a gene that'll give you your skin color. Okay, and this gene from here to here will give you, your hair color or your height, the type of height you have or the type of blood. So your DNA is so long and it has a bunch of different segments, okay, instructions, okay, for building proteins. And those proteins will determine everything about you, okay, will determine what you'll look like, will determine what chemical reactions happen in your body, that will determine your physical characteristics. So that's why these instructions are so important. They're the master plans. They need to be safely kept in the nucleus, okay, so that way they're always available to be copied. Okay, that's very important. And so now this master plan is being copied. Okay, this inexpensive blueprint, okay, called messenger RNA, okay, is a copy of that master plan. Okay, sometimes it gets edited and then it leaves the nucleus. Okay, because remember that's where the DNA is housed. It goes to the construction site, the ribosome, and the ribosome attaches to the messenger RNA, and the tRNAs, okay, start bringing these amino acids, the material that the ribosome needs to connect, and it starts to connect those amino acids to come in. And because of the anticodon codon matches, that's how the ribosome securely builds the protein that it needs. Okay, so and that's how proteins are made, guys. Okay, just like somebody would build a bike. You read the instructions, you take the parts, and you connect those parts according to the instructions. Okay. Now, Gregor Mendel is a person that we're going to learn next. Okay, and Gregor Mendel studied genes, but he didn't know what genes were. He didn't even know, okay, what genes actually did and how they determine your traits. But let's talk about genes and proteins. Gregor Mendel might have been surprised to learn that most genes contain nothing more than instructions for assembling proteins, as you just saw in the video. He might have asked what proteins could possibly have to do with the color of a flower, the shape of a leaf, the human blood type, or the sex of a newborn baby. The answer is that proteins have everything to do with these things. Remember that many proteins are enzymes, okay? They're enzymes. Enzymes are things that make chemical reactions happen, which catalyze and regulate chemical reactions. A gene that codes for an enzyme to produce pigment can control the color of a flower. Another enzyme specifying genes help produce a red blood cell, surface antigens. This molecule determines your blood type. Genes for, for certain proteins can regulate or control the rate and pattern of your growth throughout your lifetime, and also controlling the size and shape. In short, proteins are the keys to almost everything that living things or cells do. And so now, today, guys, why I'm not with you is because I'm actually getting my COVID vaccine. And the COVID vaccine is really it's remarkable. What they did is they made a messenger RNA, okay, in the lab. And in the lab, they made this guy because they figured out what proteins will help you fight the virus. And so what they did is they made these instruction blueprints, okay, so that to trick your cells into making the protein that you need, okay, to fight this virus. And so now, because I'm going to have this mRNA inside of me, my cells will make those proteins. And if ever that virus gets into my body, okay, I'll be better protected, okay, against the virus. So hopefully this helps. Translation is it's a very tricky process, but I think we've gone through it. And now it's time to learn about mutations. So see you in class.